There are roughly 8 billion of us walking around on this planet right now. We eat, we sleep, we breathe, and we bleed. And when we bleed, almost all of us carry one of just 4 basic blood types: A, B, AB, or O. Maybe you know yours. Maybe you had it written on a card at some point, or a doctor mentioned it once, or you saw it on a form before surgery. If you do know it, hold that in the back of your mind for a moment. Because here is a question that quietly sits underneath all of medicine, underneath evolutionary biology, and underneath thousands of years of human history. Why? Why do blood types exist at all? We are one species. We share something like 99.9% of our DNA with every other person on Earth. We came from the same small group of ancestors in Africa. We went through the same bottlenecks, the same ice ages, and the same migrations. And yet, right there on the surface of your red blood cells, you carry a molecular signature that is genuinely different from the person sitting next to you. It is different in a way that if a doctor gives you the wrong blood in a transfusion, your immune system will treat it like a foreign invasion and try to destroy it. It is different in a way that shaped which diseases your ancestors survived and which ones killed them. And it is different in a way that has been carried forward across hundreds of thousands of years of human evolution and is still sitting inside your veins right now. That is not a small thing. That is one of the stranger facts about being human. Yet most of us walk around our entire lives without thinking about it for a single second. Tonight, we change that. The mystery of blood types is not just a single riddle. It is actually 3 distinct mysteries stacked on top of each other, each one revealing a different layer of our existence. The first is a medical mystery. For the vast majority of human history, if someone was bleeding to death, there was almost nothing a doctor could do. Then, about 120 years ago, a scientist in Vienna realized that human blood came in different types and that certain combinations were deadly, while others were safe. This discovery saved millions of lives, transforming surgery and emergency medicine. But it raised a question that medicine alone couldn't answer. Why are these types different at all? This leads us to the second mystery, which is evolutionary. Blood type differences are not random noise. They are ancient. The blood group system is so old that it predates our species entirely. Chimpanzees have blood types. Gorillas have them too. The molecular differences we carry in our veins today were already present in primate populations millions of years before modern humans walked the earth. This means these traits were maintained across enormous stretches of time, which almost certainly means they were doing something useful, something that helped specific individuals survive the threats that killed others. Disease is the most likely answer. Pathogens, malaria, cholera, smallpox, interacted with our blood types in ways that offered survival advantages to some and death sentences to others. Over thousands of generations, these invisible wars shaped the map of blood types across the globe. Finally, there is the third layer, the history of human understanding. The science of blood did not arrive clearly and all at once. It came in pieces, through tragic mistakes and slow, painful realizations. Threaded through this is a bizarre history of myth, folklore, and cultural meaning, where societies turned biological facts into stories about personality and character. Tonight, we unravel all 3 layers. Before we dive into the deep waters of evolution, we must understand the physical reality of what a blood type actually is. Your red blood cells are impossibly small. A single drop of your blood contains roughly 5 million of them. Each cell is a simple functional worker, spending its 120-day life carrying oxygen from your lungs to your tissues. But if you were to zoom in, far beyond the power of a standard microscope, you would see that the surface of every one of these cells is covered in a complex, crowded forest of molecules. These are proteins and sugars, standing like little flags on the membrane of the cell. Some of these molecules are antigens, shapes that your immune system is hardwired to recognize. Your blood type is determined entirely by these flags. If your cells carry the A antigen, you are type A. If they carry B, you are type B. If they have both, you are AB. And if they have neither, you are type O. This is the entire foundation of the ABO system. It seems almost too simple to matter, doesn't it? But your immune system treats these tiny flags as a matter of life and death. The danger lies in your plasma, where your body produces antibodies, tiny, Y-shaped sentinels that patrol your bloodstream, constantly scanning for foreign shapes. If you are type A, your body naturally produces antibodies that hunt for B antigens. If you receive the wrong blood in a transfusion, your immune system instantly recognizes the foreign flags and launches an all-out war. The cells clump together in a process called agglutination, effectively choking your own circulation. It is a biological emergency of the highest order. So, why would our bodies evolve to care so intensely about two simple sugar structures? To answer that, we have to look past the hospital and into the ancient, silent past. Now, if these blood types were purely about preventing transfusion disasters, they would be a medical anomaly. But they are far older than any hospital. When we look at the blood types of our closest primate relatives, chimpanzees and gorillas, we find that they have their own variations of these same molecular systems. This means that for millions of years, evolution has been actively maintaining this diversity. In the brutal logic of natural selection, traits that don't serve a purpose are usually pruned away. So, if we haven't evolved toward a single, optimal blood type, it is because there is no single best type. Instead, we find ourselves in an ancient, invisible arms race. Imagine yourself not as a human, but as a pathogen, a virus, a bacterium, or a parasite, trying to infiltrate a body. You are constantly evolving to attach to your host's cells, and your host is evolving to keep you out. Your red blood cells, which were once thought to be simple oxygen transporters, turned out to be frontline warriors. Pathogens, like the malaria parasite, have spent tens of thousands of years learning to lock onto specific sugar structures on your cells. This creates a phenomenon called frequency-dependent selection. If one blood type becomes too common, the pathogens adapt to it, making that group a target. But if you belong to a rarer blood type, your cells might look invisible or wrong to the invading parasite, giving you a survival edge. Over generations, this tug-of-war prevents any one blood type from disappearing or taking over entirely. It is a dynamic equilibrium. History is written in the distribution of these types, not in stone or ink, but in the survival records of those who endured the great epidemics of the past. We are the descendants of the survivors, and our blood carries the scars of that invisible conflict. As the centuries passed, the story of blood drifted from the realm of evolution into the dark, unpredictable arena of early medicine. Before the modern era, physicians had no concept of blood types. They viewed blood as a mystical, uniform humorous fluid. When they first attempted to move it from one body to another in the 17th century, the results were essentially a cruel lottery. They tried transfusing the blood of lambs and dogs into desperate, dying patients. Some seemed to recover, others collapsed in agony. Without a map of blood types, every transfusion was a high-stakes gamble against an invisible enemy. Doctors recorded horrifying patterns. Patients suddenly spiked terrifying fevers, their urine turned dark as soot, and they died in crushing pain. They knew these reactions were catastrophic, but they lacked the framework to understand why. To them, these were not immune responses. They were interpreted as a failure of the patient's constitution, or perhaps the blood had simply disagreed with the recipient. For nearly 2 centuries, the medical community remained stuck in this nightmare of uncertainty. Surgeons were terrified to transfuse, and so thousands bled to death on operating tables, lives that could have been saved if we had only known what was hiding in those red cells. It wasn't until 1900 that an Austrian pathologist named Karl Landsteiner decided to stop guessing. He didn't have a grand theory. He simply started mixing samples from his laboratory colleagues in a grid. He observed that when serum from one person met the red cells of another, they sometimes clumped together, a process we now call agglutination. It looked almost beautiful under the lens, like tiny snowflakes forming in liquid. But in the human body, this was the moment of collision. Landsteiner's patient, methodical work turned a dark stage into a room flooded with light. By identifying A, B, and O, he didn't just solve a riddle. He created the first reliable map of our own biological identity. While the ABO system was settling the immediate chaos of transfusion, another far more complex piece of the puzzle remained hidden in the shadows. For decades, patients continued to suffer inexplicable reactions, even when their ABO groups matched perfectly. It wasn't until 1940 that scientists finally discovered a new set of markers, the Rh factor. The name itself is a historical quirk, derived from the rhesus macaque used in the laboratory experiments that identified it. But the biological story it tells is far more intimate and intense. Unlike the A and B antigens, which are essentially decorative sugars, the Rh factor is a complex protein structure embedded deep within the red blood cell membrane. Its role in human health is most dramatic not in the operating room, but in the nursery. Imagine an Rh-negative mother carrying an Rh-positive fetus. During pregnancy or birth, tiny amounts of fetal blood can cross the placenta. To the mother's immune system, that Rh-positive protein, the D antigen, looks like an alien intruder. Her body responds by creating memory cells, an immunological library that records the intruder's shape. The first pregnancy is often uneventful, as the body is still learning. But the trap is set. In a subsequent pregnancy, if the fetus is again Rh positive, the mother's primed immune system recognizes the threat immediately and floods the placenta with antibodies. These antibodies cross the barrier, binding to the fetal red cells and marking them for destruction. It is a heartbreaking biological conflict. The mother's own protective system attempting to destroy the child it sees as an invader. For centuries, this pattern manifested as a quiet, recurring tragedy. Firstborns often survived, while younger siblings suffered from profound anemia, jaundice, or worse. The breakthrough came not through evolution, but through ingenuity. Rho-D immune globulin. By injecting these passive antibodies at the right moment, doctors can mop up fetal cells before the mother system ever learns to fear them. It is one of the rare cases where we deliberately trick the immune system, preventing it from forming a memory that could destroy a life. It is the victory of clinical knowledge over an ancient, silent trap. As we look beyond the specific pathogens and the drama of pregnancy, a broader, more intricate concept emerges: pleiotropy. In genetics, this is the idea that a single gene can influence many seemingly unrelated traits. Our blood group genes are not just labels on red cells. They are master switches that affect the entire body. The same enzyme that modifies the surface of your blood cells also acts on cells lining your blood vessels, your kidneys, and the complex surface of your intestines. This means that a tiny genetic variation at the ABO locus doesn't just change your type. It potentially alters how your body handles inflammation, how it repairs tissue, and how it manages the delicate balance of clotting. Consider the act of clotting. Your blood needs to be responsive enough to seal a wound, yet fluid enough to flow through narrow arteries without blockage. People with type O blood, for instance, tend to have lower levels of certain clotting proteins, like the von Willebrand factor, compared to type A or B. For our ancestors, this was a trade-off. In a world of frequent trauma and injury, a slightly higher clotting tendency might have been a survival advantage. But in our modern world, where heart disease is a leading killer, that same genetic edge can become a liability, slightly increasing the risk of cardiovascular complications as we age. It is a classic evolutionary bargain. This pleiotropic web extends into our gut, where those same sugar structures influence the composition of our microbiome, the invisible army of bacteria that governs our digestion and immune health. Each blood type creates a unique molecular neighborhood, a specific landscape that certain microbes find more inviting than others. This suggests that your blood type is not just about what is in your veins. It is an active participant in how you digest your food and how you respond to the microbial world. We are not just a collection of cells. We are a complex ecosystem, and our blood types are the silent architects of the invisible connections that keep that system in balance. It is humbling to realize that the difference between type A and type O is not just a letter on a card. It is a cascade of effects that touches almost every aspect of your biological life. As we gather the threads of this story, we find ourselves back at the beginning. You are not just a collection of cells. You are a living archive. The blood type you carry on your medical records is a story written in sugars and proteins, a narrative that began millions of years before your ancestors ever learned to speak. It is an echo of the first primates, a remnant of the battles fought in glacial refugia, and a testament to the resilience of those who survived the Great Plagues. Our blood types have been sculpted by 1,000 different forces: the bite of a mosquito in a malarial swamp, the contaminated water of a medieval slum, the random drift of small migrating bands, and the silent, deadly negotiations of pregnancy. We have learned that blood types are not personality labels or destiny markers. They are, at their core, biological compromises, a ledger of survival in an unforgiving world. The reason they still persist, balanced in different proportions across every continent, is that the human story has never been one of a single, simple path. It has been a branching, messy, wandering journey through environments that demanded different skills from our cells. Sometimes, being type O was a shield, other times, a liability. Sometimes, A or B provided the subtle advantage that meant the difference between leaving descendants and vanishing into the dust. So, the next time you see that letter on a card or a screen, remember, it is not just a medical fact. It is a link to the deep past. It is a bridge between the clinical reality of an operating room and the ancient struggle for survival that defines our species. We are still in the middle of this story. New tools are allowing us to sequence ancient bones and read the genetic code of those who came before us, and we are only beginning to understand the full complexity of how our blood types interact with our lives. But for now, you can hold on to this. You are carrying history in your veins. Every heartbeat is a reminder of those who came before you, those who faced the same world, survived the same challenges, and passed their blood down to you, an unbroken, living chain stretching back into the mist of time. And that, in its own silent, remarkable way, is enough.